Welcome students. Today I have come to clear all the details about the antioxidant assay and antioxidant determination of antioxidants and the calculation of IC50. Last class even I was not very clearly explained about the Excel values, how we can determine the line equation. So we will see today very clearly. But today what we are going to discuss is about the antioxidant in plant extracts. After extracting, we have taken an extract of ethanolic extract from this Melia dubaya, that is Malabar Vembu, and also it is also called Malavembu. That other one is different actually. This is different from this is Melia dubaya. The other one which I told the name for this one is different. The seed, there is a lot of work have been done by the leaves, but we have taken the seed, very hard seed, crushed, and the powder have been extracted, and then we have seen this antioxidant effect. And this last time we have discussed about the ferric and ferrous antioxidant effect by means of using ferricyanide. But here today we are using the technique which we have taken is nitric oxide the radical by means of using coupling reaction. Even we are just in the experiment itself, I was just given some chemistry background, how the coupling reaction and the calorimetric method, how it plays for the determination of antioxidant activity. And this is an very interesting experiment which can be performed by any researcher, and also in degree level project most of them are carrying out. So I hope it will be useful for the degree pharmacy students and who is performing the determining the activity, antioxidant activity. And we have detailed, I have told about antioxidant in the previous class. Now only the experiment. And here the nitric oxide NO generated from sodium nitroprusside, SNP is the symbol we have used here, sodium nitroprusside, was measured according to the method of Markov K or Markov saying in et al. A group of people have published this paper in 1997, have taken as a reference for this study. And here the materials required is distilled water, anhydrous sodium phosphate dibasic, sodium chloride, anhydrous sodium phosphate monobasic, sodium nitroprusside, sulfanilamide, phosphoric acid, and 1-ethylene diamine. For coupling reaction, we need the color formation for the determination. Centrifuge tubes also needed in case sometimes for centrifugation, but here we are not much using it. Micro pipettes and bulk pipettes and volumetric flasks and test tubes with marked properly, cleaned, and a water bath and other glasswares which were needed in the laboratory, and UV spectrophotometer for us to find out the absorption of the standard and the test and the control which we are going to deal with. So we are starting with the reagent preparation also, because there are ready-made reagents available, but still reagent preparation is needed. The student have to not to depend on the ready-made, and we should know how to prepare if suppose we have the chemical. This is phosphate buffer saline pH 7.3, weigh 10.9 grams of anhydrous sodium phosphate dibasic Na2HPO4, 3.2 grams of anhydrous sodium phosphate monobasic NaH2PO4, and anhydrous of dibasic and monobasic. Instead of we are doing here, that is Na2, here it is NaH. You can have an addition of 90 grams of sodium chloride and dissolve in 1 liter of distilled water. Adjust the pH to 7.3 and make the solution up to final volume 1 liter. If possible, you can prepare 2 liters also, because for dilution it may be needed. If you need, we can make it, because we have taken a blank by means of only this buffer have been used as a blank for us. And sodium nitroprusside 5 mm, that means millimoles. So we are talking about moles per liter. Molecular weight is very important for preparation of this solution. So students should know, when you see small m and m, it means millimole in phosphate buffer. In the phosphate buffer which you are prepared have to be used for dilution. pH should be 7.3. So molecular weight of sodium nitroprusside is 297.95. So I made this calculation for you to understanding, that is, if you are 1 mole solution, if you want to say, we have to weigh 1 molecular weight, that means 297.95 grams of sodium nitroprusside in 1 liter of distilled water. Then we can say, for distilled water, or here we have to use the phosphate buffer, then we can say it is 1 molar. But we don't want 1 molar, we want 5 millimole solution. So we have to calculate according to that. The concentration of 5 millimole is the same as 0.005 moles per liter. That you know that 5 millimole means it is nearing to 5 microgram. So it is 0.005 grams. So take 0.005 grams means that's moles. So what is this moles? That is, you have to see that how much grams should be taken for 0.005. 0.005, when you multiply this molecular weight, then you will get 1.4897 grams. So from the molecular weight, each and every moles will be different according to the molecular weight of the components. So we have taken sodium nitroprusside, the molecular weight is 297.95. That's why we have to weigh 1.48975 grams, dissolve in 1 liter, then you will get the 5 millimole solution of sodium nitroprusside. I hope it is very clear. If it is not, you can just write down and you can note it down. It's not a very simple thing which you have done in your analytical chemistry. As we need the solution in phosphate buffer, saline, so we have to use for dilution the phosphate buffer which you have prepared, and weigh 1.48975 grams of sodium nitroprusside and dilute with phosphate buffer saline pH 7.3 and make up the volume up to 1 liter, so that your solution is ready now. So this is firstly you have to prepare for an experiment. And Gris reagent is a reagent which is very much interesting for this experiment, especially nitrile experiment. So here in Gris reagent, ready-made reagent is available, but we should know how to prepare also, because ready-made can be preserved and also freshly prepared will give you good results. So 1% which contain 1% of sulfanilamide in 5% of phosphoric acid and 0.1% of naphthalene ethylene diamine dihydrochloride. This is the grist reagent. So preparation of grist reagent, mix equal volume. Just a minute, it is just I have to pull down and so that you can see clearly. So you see here, preparation of grist reagent, mix together with equal amount of N1 naphthyl ethylene diamine. That is a component A, and component B you have to take the sulfanic acid to form a grist reagent. Prepare sufficient reagent immediately when your experiment is started. You have to freshly prepared is very good. Ready-made reagent is also available. So most of the students might have gone for the ready-made reagent, I hope that. So even it is also possible for you to go for this experiment. And nitric oxide NO release from sodium nitroprusside as a strong NOH plus character, which can alter the structure and the function of the many cellular components, even plant extracts. So plant extract or a standard antioxidant which you are using, ascorbic acid, will exhibit NO scavenging activity, inhibiting activity, reducing activity, leading to the reduction of nitrite concentration in the assay medium. So nitrite is very much needed for your complex formation in this assay and to form a disodium salt and to give a very good colorimetric color which can be affected by means of this scavenging effect. So that's why how much it can have a reducing effect or antioxidant effect of the plant extract, and the standard can be compared by means of this IC50 value. We can predict this antioxidant activity. That is the main understanding. If you know this finished, that is a very simple experiment. Even it can be conducted in the normal lab also. I expect in a degree level student have to do this experiment. Then the NO nitric oxide radical thus generated react with oxygen to produce a nitrite ion. Under aerobic condition, NO reacts with oxygen to produce a stable products, nitrate, you might know nitrate, you might have studied NO3, NO2. So nitrate and nitrite, NO3 and NO4 can be produced in aerobic condition, oxidation. So that can be prevented by means of antioxidant. So you know what is the problems by means of radicals in our body, how it is affecting. This last class we have explained about what is the use of antioxidant effect of various fruits and plant sources, which is giving more advantages in various ailments in human being. So that you have to go through what is an effect in various ailments that is important, like cancer, cardiac diseases can also be prevented by means of using this antioxidant in a food, and also various drugs can be applied in this field. And the quantities of which can be determined using grist reagent, scavengers of nitric oxide compete with oxygen, leading to reduce the production of nitrite ion. So this will have, if your extract is having more scavenging effect, automatically it will reduce the NO production, NO radical, so that you can see the depth of the color or the depth of the disodium salt production can be inhibited. So that is why we can find out, that's how we are finding out what is the antioxidant effect of my plant extract. So here we have taken the seeds of Melia dubaya and we have extracted by means of continuous oxalate method, and we have taken various dilutions and we have made the test with comparing with the standard of ascorbic acid. So I have given you detail about this small information. I hope it will be helpful for the student. And this is the reaction which is taking place. See, if the first one is sulfanilamide, which react with the nitrite which is produced from here. We are using the nitrite will be produced by, that is sodium nitroprusside, which can produce and react with this sulfanilamide, and this will be inhibited by the power of the antioxidant effect by your standard and by your sample which you are using. Understand? And then it will react with the, and it will first form the diazonium salt, N2 plus. You can see in the structure diazonium salt, and this diazonium salt we are adding the, it will just react with the reagent of N1 naphthalene ethylene diamine, and coupling will take place, and you are getting azo dye. You will get the azo dye which forms a pink or a red purple color, which can be easily identified by calorimetric method of in 540 nanometer. Even you can go for a medium concentration by means of lambda max finding, and then you can go for absorption calculation. But already the researchers have identified and they said that 540 nanometer is very much feasible. So in case if you want to do is very perfect, you can go for lambda max estimation or scanning estimation can be done, because high sophisticated instruments are nowadays available in every institution. The Griff's reagent, that is what the Griff's reagent which you are using, which contain this reagent, will make reaction with the diazonium salt which formed by the sulfanilamide which is present in the Griff's reagent. So Griff's reagent is a very important reagent in this experiment. So Griff's reagent involves two subsequent reactions. When sulfanilamide is added, the nitrite ion reacts with the Griff's diazotization reaction as shown in the reaction down to form a diazonium salt, which then reacts with N1 naphthalene diethylene diamine in an azo coupling reaction. Understand? So forms a pink red azo dye. Sometimes it is purplish because of very low quantity of nitrite, and react with the disodium salt. So which can be absorbed by 540 nanometer in our spectrophotometer, UV visible spectrophotometer. You can see this is one type of calorimetric method which can be easily determined for the antioxidant activity of a plant extract, and even it can be used for various physiological function can be also determined. It become a very good interesting experiment which can be done for various research. That's why I took this topic for giving you the explanation. Now we'll go for the method. We have taken these seeds actually, Melia dubaya seeds, and we crushed it very hard, dried, crushed with hammer, and then by means of blending machine and mixers, and then it is powdered, and very difficult, but it's very much powdered, and we use for continuous oxalate method by means of hot percolation using oxalate method using ethanol. We have got the so we have taken ethanolic extract of Melia dubaya seeds. Standard we have taken ascorbic acid. So how you are going to prepare the dilution? First we will start with the sample Melia dubaya extract which you have taken. Yield have been already calculated. That is a different experiment which you have done, and we take 250 milligram of Melia dubaya seed extract, that means especially I missed that is ethanolic extract, which is added to the standard flask, 250 ml of standard flask as shown in the picture. And so that what will be make up the volume with ethanol with 1000, and it will be, if you see the calculation, it is 1 milligram. See, because 250 milligram in 250 ml, 1 milligram per ml is the concentration, or in other words you can say 1000 microgram per ml. And from that we are removing in different dilution, we are making it. So for example, we take 2 ml. So what will be? 1 ml will have 1 milligram. So 2 ml will have 2 milligram. 2 milligram in 100 ml, make up the volume with ethanol. So 2 milligram divided by 100, automatically you can see that it's 0.02, so that it comes 20 microgram per ml concentration. And same thing, 4 ml, you will get 40 microgram per ml concentration. And the next, if you take 6 ml from the stock solution and make up the volume with ethanol, 60 microgram per ml concentration. And you take 8 ml and make up the volume with ethanol, 80 microgram per ml concentration. And you take 10 ml from the stock solution and make up the volume with ethanol, you get 100 microgram per ml concentration. So this is a sample which you have prepared by means of ethanolic extract of Melia dubaya seeds. And the next one, standard, also we have to use these dilutions properly. So what you are going to perform is 200 milligram of ascorbic acid you have taken as a standard in 250 ml standard flask, and ethanol as a solvent, make up the volume. So the same thing, 1 milligram per ml, or you can say 1000 microgram per ml. And from that, various same dilutions are made. So when you take 2 ml, 20 microgram per ml, 4 ml, 40 microgram per ml. Make up the volume in 100 ml standard flask, and 6 ml, 60 microgram per ml concentration, and 8 ml, 80 microgram per ml concentration. And you take 10 ml, you are getting 100 microgram per ml. This is the dilution which you made for a standard ascorbic acid. So this is the 2 slides which I have explained for the standard and test sample we have prepared, or test you can say. Sample or test you have taken for each dilution which you have prepared, that is, you have taken 20, 40, 60, 80 and 100 microgram. So from that 1 ml, draw 1 ml in a test tube and add 1 ml of sodium nitroprusside solution which you have prepared, and incubate for 2 hours in 30 degree centigrade plus or minus 0.5 degrees centigrade in an incubator, and then after incubating 2 hours, add 0.5 ml of grease reagent. Then immediately you will get the color formation. So this is just I showed a test tubes which they are using. And standard also, you do all the dilutions with 1 ml of sodium nitroprusside solution, incubate it for 2 hours with 30 degrees centigrade plus or minus 0.5 degrees centigrade, and plus 0.5 ml of grease reagent, and you will get the formation reaction diazonium, that means the salt formation, diazonium salt formation, and then azo formation according to the, and you can keep, this is the incubator picture which I want to show you. And you need a control also. What is control? Control is different and a blank is different. Control, blank is for the reading, absorbance. Here control is also needed. Control, no sample, that means no extract dilutions, no standard, only you take, if you want to be more perfect, you can take 1 ml of ethanol, because you have done dilutions for both standard and sample with ethanol. So 1 ml of ethanol which you are doing for dilution you can take, and add 1 ml of sodium nitroprusside incubated at 30 degrees centigrade, and for 2 hours you have to do the same work for this one sample or one control, and add a point after incubating for 2 hours with 30 degrees centigrade plus or minus 0.5 degrees centigrade, and then add that control test tube with 0.5 ml of grease reagent, so that you will get the control color. This is the main important fact. 3 things you have to remember. So now we are going for the absorbance. The absorbance of chromophore, purple azo dye, there we said red pink, it will be like slightly purple, formed during the diacetation of nitrite ions with sulfonamide and the subsequent coupling with the N-naphthalene ethylene diamine dihydrochloride was measured in 540 nanometer. The nitrite generated in the presence or absence means control, presence of seed extract or the standard estimated using, you can by means of standard curve for a ascorbic acid, and we can find out what is the concentration of the unknown sample is also possible for you to determine. That every student may be knowing by means of graphical method it is possible. Understand? So that is also possible for you to determine. So this is just a picture to show you of a purple color. Sometimes the color may be different according to the reagents which you prepared and used and the sample of extracts which you have applied. Absorbance was measured at 540 nanometer using UV spectrophotometer. A high, suppose Simashu is the one which we have used for UV 180 UV visible scanning spectrophotometer. Various samples have been prepared, and phosphate buffer saline pH which you have prepared and used for various dilutions and adding from the beginning for the experiment should be used as a blank, because we need a blank for the instrument to find out the absorption of any solution which you prepared. So blank should be used. So don't get confused with the blank and the control. Control is different, the blank is different. Understand? For control measurement also you need a blank with phosphate buffer saline pH of 7.3 which you have prepared can be used as a blank for here. And now we can see this ascorbic acid absorbance from the instrument which you did is for 20 microgram concentration we got 0.253, 40 0.197. So the absorbance is coming down for the standard. Why? The action of the antioxidant action of the ascorbic acid to the nitrile and the scavenging, reducing the amount so that the intensity, the formation of the azo have been reduced. That's why the word scavenging is used. Understand? So you can see what is the percentage of inhibition. How will you calculate? Percentage of inhibition is equal to 1 minus absorption of the control, which is we have seen. Control we have seen without adding standard or the sample 0.327 have been shown because it's control. There is no antioxidant effect. So directly the color was so intense, 0.327, you can see very clearly the absorbance which you have got. So how the experiment I have shown, very interesting experiment with a good result. And 1 minus absorbance of control divided by absorbance of standard into 100 can be used, or you can use this type of also, because by means of 1 instead of using absorbance of control minus absorbance of standard divided by absorbance control also into 100. Back bracket, don't forget bracket. Last time I forgot a bracket in my explanation. It made a lot of difference in the calculation. So this is how they found out the percentage inhibition 22. It is 0 for control because you know that 0.3 2, 7 is the control value, 1 minus the same, it will become 0 automatically when you multiply. 0 when you multiply with any number, it will become 0. That is why for control we have taken as 0. Understand? So the inhibition for control is 0 because there is no antioxidant is added. So we are going to see the antioxidant inhibition effect of the standard and the sample comparison. This is standard. So now we need the IC50 calculation. So we need another one for sample also, the same dilutions IC50 and standard also for our comparison. First this one I have done for standard. And what is IC50? That antioxidant activity was expressed as IC50 and compared with the standard, the half maximal inhibitory concentration of IC50. It should be in mathematical expression, and it is easy for you to calculate through Excel. So I want to show you this Excel, how to do and how to do. Very, very simple. I hope last experiment which I explained was not well clear. When I saw it, it was having some bracket I left in the Excel so that it made less value of IC50. So the students can, now they can clear the doubt and they can experiment with their own laptops, their own Excel calculation can be done. So let me go for the Excel and let me show you how to calculate now. So it will be easy for you. Instead of going to the next slide, directly we can have an Excel. So now I have opened an Excel file and we are taking a new sheet in which I hope it is visible for you, the screen. See now, let me go to the reading. First we have taken this is the reading which we have made. Coming to the Excel here, just I am taking some place here and just pasting it. We can paste, and even this we can make it like this. What is this? This is concentration. Okay, 20 microgram concentration, and then we can go for the sample reading, sample absorption reading, which we have done here. Not absorption, the percentage inhibition calculation which you have already done for each concentration. Okay, plus copy, and I am just pasting here. This is the, sorry, I made it, we can paste it by means of this one, so that, and also we can make it center. This is the, even I can bold it, or you can, the size can be increased and bold it. See now the inhibition, you can see here the sample inhibition. Inhibition, I have just put short form inhibition. So this is the two reading which we have got, and we want to find out the IC50 for our experiment. So it's very simple for you to do. So what you have to do, Excel, plain sheet, and the concentration, sample reading. Even for standard also you can do the same thing. So now you just go to the insert and come to the graph. There is two types are there. Don't go for this one, come for this one and select the second one. There is a scattered with smooth line and markers. That is scattered with smooth line and markers. Select that. It's a plain screen you got. Am I right? And now you go and keep the cursor here and right click and select data. Select data. You get another box as shown in the picture, as shown in the screen. And now click add. You will get what is the series name. You want to write the series name, you can write the series name. What is the name of the series they are asking? So we can write it is sample, that is Melia dubai extract, the plant extract. And then you can go for x-axis. What do you want to do? Keep your cursor there and then come to this place. This is x-axis. Select. See, automatically it has come to there. And then you come to the y-axis. There is, they put one equal to bracket will be there. Just you have to go for delete that. And then now y-axis you have to select this place. Now you got the graphical expression itself for sample directly. And now still more the work is there. The line equation you want to find out. So you put OK here and then go back to the place. Here you can click and release it. And now keep the cursor here, click. Add. What you should add? You should add data in the label or axis. Axis title is very important. Y-axis, just click it. Y-axis, what you have to write? Y-axis, it is of inhibition. Inhibition. Okay? And then come to this x-axis. What should be written? Concentration. Concentration. That is mcg per ml. We have used, that is the concentration. Click here. It is finished. Now again click here and add. Now we are just making it to be proper way, just naming. See, sample graph we have got. X-axis we have made into concentration microgram per ml. Oh sorry, yeah. Y-axis is inhibition. Now just you go for trend line. The trend line, there is an arrow. Click there. More option. And immediately it comes here for more option. It should come for trend line, more option, and then you have to take it down. Actually it should come down here. You can see there is a display this side, because you can see my screen below, it has been shown automatic, customize, forward, and here you can see display equation chart. See there, immediately displayed y is equal to, and you can show display the r value, r square value of the graph of the line which you got. So both of them, y value and r square value, just you can display it. May not be seen very clearly, so I am just enlarging it for students to see my screen. See here, this is what I got from the graph actually. I am just pulling it, not pulling it up here. Sorry, shall I pull it out? It may not go out, only it will be kept here. So you can see that is y is equal to 0.804 plus 7.64 and r square value is 0.9963. So students should know that what is this r square value. It very important. It should be 0 to 1. So our experiment is whether it is moving smoothly or not. It should not be 0 to 1. Sometimes it's y value plus 7.68, maybe sometimes minus also it is possible, but we talk about y is equal to 0.804x plus 7.68 is our experiment's line value. Understand? Now, what is the formula? From the formula we know that y is equal to, from the graph itself, that 0 point value is actually, it is the m value and plus c. That means what we can say, m x plus c. We got the formula. Now we have to find out the formula. X is equal to, very small mathematical expression. X is equal to means when you keep the x here and take it out, so you can see x is equal to y minus, it will be the answer, y minus, that is bracket. Last time I forgot to put the bracket. You should put bracket, otherwise the formula will be changed. Y minus c bracket close divided by m. That is the x value is the, when you take y as a 50, you will get 50 x value. X is concentration. So we are going to find out the maximum of half concentration of the activity of the antioxidant activity. So if you want to make it y 50, you will get 50. If you make it 90, you will get 90. Understand? Even that is the main aim of finding out x value. So you can check it now. Even if the student, if they are, you are thinking that what is the this value, whether it is right or wrong. If you want to know about that, this formula which I have taken from the graph only. So if you are having a doubt whether this formula x minus c divided by y is the correct, just calculate and see. For example, x is equal to, for example, if you want to see x value, what is the concentration which the absorbance you have got. If you see, you got somewhat 40.433 in the 40 microgram concentration as an inhibitory in the sample. So you know the value, so you can see x is equal to y is the, what is the y value is the absorbance, and c divided by m, you should get the proper concentration. And that is the formula, the line length. We can check also whether you got this 40.43 is correct. If you want to apply the x value for, it will come 100% sure. Understand? Y value we know already, it is there, and when we minus it by the concentration and divided by the m value which is got from the calculation. So we can make it here for our IC50. X we are going to find out now. This you can put x here, and the second one you can put y, which we are making as 50. We are going to find out 50. And the next one is you can make the C value. Understand? C value. What is the C value? 0.8044. And next one is your M value. M value. Just I put capital M value. M value is actually, what is the M value from the equation which we got? 7.68. So again, for your visibility, I'm just making it little bigger size so that the student can see it little more clearly. Understand? So this is the value which we got, and I'm just making it center also so that you will be clearly you can see that what is the value which we are given. So this is what we have to find out now. So how to find out the value, x value from this formula? From this is the formula, x is equal to, because we know this is the formula which you got from the equation which they have given. Y is equal to mx. This is got from the calculation from the Excel itself. We got y is equal to mx plus c. From that we found out the equation x is equal to y minus c by m. Y minus c bracket we have to put without forgetting, because the whole divided by m it is. Understand? So that will give you the formula. And so that now I made a small mistake here. This is the formula, that is mx plus c. 1 minute. Here, that is the mistake. When you do mistake here, everything will be wrong. Actually, m is equal to correct cm, mx plus c, y minus c by m, and m value is the last one actually. So here you know what is the mistake I have done here. You can just, this type of mistake when you may also do, because we told y mx plus c, so this is the c, so the value I changed here, this is not the c. Actually c value is here 7.68 and m value is 0.8044. Sorry, 0.044. I hope you got the mistake which I have done. See, from the equation, this is how. And now we have to calculate here. This formula should be applied. 50. How to apply? First you have to put equal to, because fx you are going to do equal to, and what you are going to do is bracket. You have to start the bracket, and then you select this y. Understand? Y you select, and you make it minus. You put one hyphen, minus, which one? C. You have to minus c. Automatically it is Excel, no? So automatically it will calculate, and bracket close, and put a slash, that is divided by which one? M. Divided by m. Can you follow? And now if you put enter, you will get the value of 52.62 is the value of y value when you put 50. That is why it is called IC50. Understand? So this is how we are calculating. For example, IC50 is 52.62 for the sample. That means the intermediate 50 concentration of inhibition of antioxidant effect is 52.61. You see here, I put the formula exactly. Even if I put change 90, you will get 102.3. So the activity value, you see how it is changing. So that is a very, very important factor in this place, which we can make it in Excel. Very interestingly we can find out. Even for the sample standard also, go for this data, and it is possible for you to do very easily. Understand? Can you follow? This is what today I am very happy that I have given some information which can be calculated easily by this Excel. Now we'll go to our explanation part, the values which can be calculated already. So this is what which just now we calculated, and already I did the same with the Excel for inhibition. The formula you can see how we calculated for IC50. So I hope, please try to do once by means of your Excel laptop. One, you try whether it is right or wrong, which we added in our experiment. Last class I told a mistake actually. I did not put the bracket when I was calculating. So the value came down. Now 90 can be calculated, 50 can be calculated, half inhibitory and 90%, 70 or 80. All these things can be calculated very easily by the line equation which you got. And R square value, what is the use of R square value? The student may be thinking, why we want to go for Excel R square value? R square value we did not use for calculation, but it shows it should not be more than 1. It should not be less than 0. It should be in between 0 to 1. Understand? Can you follow? That is important line value. That's why experiment is going in a smooth manner. So it gives, whenever you are doing a research, it is necessary for you to go for the error function. So now for the same thing for excuse me, so you can see here, and afterwards you can see the coming for the next step of test. This is for sample of the extract dilution which we made. I made all the calculation for percentage inhibition as we are shown in the standard. For ethanol extract of Melia dubaya seeds, we got the value is 50.1423, little less than the standard. Standard is 52. In 50%, it is called 50. So you can see this is, both of them have been compared. The IC50 value, the standard is little higher. So sample is giving very good antioxidant effect, seed extract, when compared to the standard. So it's a good sign of a natural extract which gives you an antioxidant effect. So the inhibition of nitrate formation by the plant extract and the standard antioxidant ascorbic acid were calculated relative to the control which you have used, and inhibition data percentage inhibition were linear against the concentration of each extract and standard antioxidant. IC50, which is an inhibitory concentration of each extract to reduce 50% of nitric oxide formation, was determined. Ethanolic extract of Melia dubaya seeds is having antioxidant activity by scavenging the nitric oxide free radical. This is what the interesting experiment. Thank you very much for being with me. I hope this will be useful for your experiments.